Antidiuretic hormone regulates the retention or excretion of water by the kidney. The events are in the collecting duct of the nephron, the loop of Henle, and here the collecting duct. Collecting duct. These cells have receptors that bind ADH. Aquaporins are always present on the basolateral basement membrane side of the cells lining the collecting duct, but aquaporins are not present in the apical side unless ADH binds. If we looked at one of those cells, and basement membrane is on this side, this is the basolateral side, and basement membrane would be extending all the way along here, this mesh of proteins. Here's the lumen of the collecting duct with the developing urine. Aquaporin 4 is always present in the basolateral membrane. ADH, or antidiuretic hormone, binds to a G protein coupled hormone receptor, and when it binds, subunit of the G protein activates adenylate cyclase to convert ATP into cyclic AMP. That cyclic AMP then activates a protein kinase A. Protein kinase A phosphorylates aquaporin 2, another aquaporin. And aquaporin 2 is normally held in vesicles that are in the cytoplasm. Phosphorylation and these events cause these vesicles to fuse, which moves the aquaporins here into the membrane. Remember that the concentration of urine at the beginning of the collecting duct in a human may be something like 60 or 100 milliosmoles in comparison to the approximately 300 milliosmoles of blood or extracellular fluid. When aquaporin 2 is in the membrane, that allows water to move into the approximately 300 milliosmol cell cytoplasm, which changes the concentration here to higher and higher concentrations. As water moves in, that dilutes the solutes in the cytoplasm. That water continues out through aquaporin 4. At the beginning of the collecting duct, where the extracellular fluid is around 300 milliosmoles, that allows water to move out until we approach the same osmolarity in the developing urine as in the extracellular fluid. As we move down the collecting duct, we go from 60 here to perhaps 300, and this would be the cell I just talked about. As we go deeper and deeper into the medulla, we change from 300 milliosmoles to 500, gradually increasing down to something like 1400 milliosmoles. And all along here, if ADH is present, water continues to move out of the developing urine, and so the deeper we go, we get closer to something like 1200 or 1100 milliosmoles. We don't get all the way to 1400 because even if that high concentration can sometimes be developed in the medulla, of course all this water coming out winds up diluting it, and the best we manage in a human is somewhere not that far above 1000 milliosmoles.